Hello everyone, this is our developmental biology series for CSIR-NET exam, and today I will discuss how the anterior and posterior pattern formation occurs in Drosophila. In Drosophila embryo, we know that there is a syncytial specification occurs, and here the many genes play important roles for the anterior and posterior determination, right? So major important genes here we can see that the how the major question of this topic today is how anterior and posterior axis determination occurs in Drosophila. In Drosophila, now major genes are the bicoid and nanos. We know that when we retain the gene, in the gene will be small letter, and proteins will be in capital letter. Bicoid, in if it is written like bicoid in capital letter, these are for protein and these are for bicoid gene. Now major important part is here. Now bicoid get translated and form concentration gradient at the anterior side. Actually bicoid present in the bicoid actually form the gradient in the anterior side, and nanos form the gradient in the posterior compartment of the Drosophila embryo. Now, here important is that Oscar mRNA with the help of plus N directed motor protein, like kinesin protein reach to the posterior site get translated. Bicoid mRNA will reach to minus N with the help of minus N directed motor protein, that is dynein. Dynein is a minus N directed motor protein and kinesin is a plus N directed motor protein. These two protein play important role. Now the Oscar protein traps nanos mRNA. Oscar protein trap nanos mRNA. Oscar. Now this so large number of nanos mRNA get associated at the posterior end. So here is that Oscar mRNA. Oscar mRNA helps to get the local Oscar mRNA with the help of plus N directed motor protein reach to the posterior site and get translated, and bicoid reach to the anterior side with the help of dynein, that is minus N directed motor protein. Now the anterior forming genes, major important genes are bicoid. The mutant phenotype, if the bicoid there is a mutation, head thorax deleted, replaced by inverted telson. Its function is graded anterior morphogen contain homeodomain and repress the caudal mRNA. Actually bicoid repress caudal synthesis, or bicoid, if the bicoid present, they actually inhibit the posterior formation or tail formation. Now in exuperentia, anterior head structure is deleted. If mutant exuperentia is deleted, anterior head structure is deleted. Anchors bicoid mRNA. Actually bicoid is the major one, and these two protein also play important roles. This is the secondary one. Major one is the bicoid, which play important roles in the anterior or head formation in the Drosophila embryo. Now we'll see the how the bicoid replacement experiment, how if we exchange the genetic combination, if we replace the bicoid in different regions of the Drosophila embryo, then there different developmental patterns can be produced. Now we'll see that experiment. So experiment, first see that first experiment. So if we in an early egg, Drosophila egg, now puncture the anterior part and let anterior cytoplasm to get ooze out. Anterior cytoplasm. Now what's the no head formation? Because here bicoid gradient is lost. Bicoid, this morphogenetic gradient is lost. So as that, for that reason, no head formation occurs in the early stage of the early egg. Now in case of second, the puncture at the posterior region allow the posterior cytoplasm to ooze out. Now if we remove the posterior cytoplasm, then also the nanos mRNA get lost, so no posterior formation. Now third experiment, if you see the inject bicoid in posterior site, if we inject bicoid here, anterior site is normal, remain normal, The bicoid is here normal, but what you are doing, you are injecting bicoid in the posterior side in case of, and also removing the posterior cytoplasm. After removing the posterior cytoplasm, you are injecting the bicoid into the posterior region. Inject the bicoid in the posterior side. Bicoid is a strong inhibitor of nanoons and head formation at the posterior side. It will show the two head formation, that is called dicephalic mutant. So bicoid gene mutant, no bicoid mRNA, so no gradient of bicoid on the anterior side, no head formation. So bicoid is absolutely necessary for head formation. So bicoid is absolutely necessary for head formation. Now see that another experiment here, we can see that inject anterior cytoplasm from a wild type embryo. If you inject the bicoid mutant and you are injecting the anterior cytoplasm in the bicoid mutant, then what you are observing, the normal development, normal head formation will occurs, because here bicoid gradient is maintained. Whatever we are injecting, that's actually the cytoplasm contain bicoid mRNA, and bicoid mRNA get translated and form bicoid protein, and that's why their normal head development occurs. Now here we can also see that this experiment, first see that these are normal development, wild type bicoid mRNA is the anterior side, so acron, head, tail, abdomen, and tailson formation. So head to tail formation occurs. It's completely normal wild type development, right? Now second in experiment, you can see that development of second experiment is the development of bicoid deficient mutant. Development of bicoid deficient mutant. These are bicoid deficient means bicoid gene is non-functional here. So here two tail, both, and there is no anterior side, both side tail formation occurs, because the posterior nanos mRNA it can take up the function of the anterior side. So as the bicoid is deficient, bicoid is lack of, so both side tail formation occurs. Bicoid deficient phenotype have two tail, that is both side two tail formation occurs. Now third experiment, you can see that add to anterior. Third experiment, if you see, third experiment, add to anterior end of the mutant. If you add anterior end into the mutant, there is a development of acron, head, tail, abdomen, and tailson, right? So head to tail formation occurs, completely normal development. Now what you are doing, now you are injecting bicoid into the middle of the mutant. These are also BCD mutant, and in this mutant you are also, these are also BCD or bicoid mutant. This also, this egg also bicoid mutant, and you are injecting BCD mutant, bicoid mutant, and you are injecting the bicoid into the middle of the mutant. You are injecting bicoid into the middle of the mutant. Now what will be the observation? If you inject the bicoid in the middle of the embryo or the middle of the mutant, then tailson, these are tailson formation, these are tailson thorax, head, tailson, abdomen. That means tail, head, tail formation occurs. So head in the middle, head in the middle it is formed. So actually it is indicating, what is the indicating, that if you inject bicoid, that's actually for head formation or anterior formation. Now in the another last experiment, you can see that add to the posterior cytoplasm, posterior end of wild type embryo. If you add bicoid, add bicoid to the posterior end of the wild type embryo, then what will be happen? So you can see that observation, here acron, head, tail. Now here is abdomen, these are thorax, acron, head, thorax, abdomen. then thorax, head, abdomen, thorax, head, anterior. So both side two head formation occurs. Both side two head formation occurs. That is, two head formation takes place. So if in summary we can say that the bicoid and nanos are play important roles for anterior and posterior axis determination of Drosophila. So in quick review we can see that first what we have seen, how the anterior posterior determination occurs in Drosophila, how Drosophila embryo develop, how anterior posterior axis determination occurs in Drosophila. Then we have seen that bicoid gene, bicoid mRNA, bicoid protein. Then we have seen that Oscar mRNA plays important role for localization of nanos and the bicoid mRNA. So Oscar actually, now a major important player is bicoid. So bicoid actually anterior head formation is a bicoid. These are important one. And then second we have seen that experiment. So in this experiment, first is if you remove the anterior portion, no head formation occurs. That means bicoid morphogenetic gradient is lost. So no, that's why no head formation occurs. If you remove the posterior part, so no nanos is there, so no posterior formation takes place. If you remove the in the third experiment, if you inject the bicoid into the posterior side, then bicoid is a strong inhibitors of nanos. These are very important term. Bicoid is a strong inhibitors of nanos and its head formation at the posterior side, in the where actually tail should be formed, there will be the head will be formed, right? So bicoid gene mutant, no bicoid mRNA, so no gradient of bicoid on anterior side, no head formation takes place. These are now last one we have seen the different experimental view. So now if you inject different types of different mutant form of bicoid and wild type embryo, then there is a different developmental patterns occurs in the Drosophila. In the next lecture we'll discuss the how the Drosophila segmentation occurs, Drosophila body segment patterns, how it is developed, and different genes and their signaling patterns in the development we'll discuss in our next lecture. Hope that's make helpful. Thank you.